Welcome back, dear students. In today's video, let us discuss one of the most important topics from second PUC syllabus, that is central dogma in molecular biology, and it is the topic from the chapter molecular basis of inheritance. And let us exactly know what central dogma explains us. In molecular biology, central dogma explains the process of protein synthesis, and central dogma in molecular biology was proposed by Francis Crick. In very simple terms, if you ask me, what is central dogma in molecular biology? Central dogma always explains the direction of flow of genetic information in biological systems. As we all know, DNA is the master molecule, whether it is a prokaryotic cell or eukaryotic cells, with very few exceptions. In most of the living systems, what molecule acts as genetic material? That is DNA. As we all know, DNA stores the genetic information in the form of nitrogen base sequence, otherwise called nucleotide sequence. So, this nucleotide sequence present in the DNA controls all the activities of the cell. So with this it is very clear, DNA is the master molecule which stores the genetic information and it controls all the activities of the cell. And one of the most important activities happening inside the cell is protein synthesis. So at which point of time, which type of protein has to be synthesized? It is always decided by DNA only. But DNA cannot come out of the nucleus, because as we all know, proteins are synthesized on ribosomes. So with this it is very clear, at the time of protein synthesis, there is a first step which is called transcription. So simply speaking, what is transcription? Synthesis of a single-stranded RNA from double-stranded DNA. This process is called transcription. And it is followed by what? Translation. So here I have represented. What is translation? Synthesis of a protein or polypeptide from an RNA on ribosomes is called translation. So with this it is very clear. In biological systems, protein synthesis always includes what? Two steps, that is transcription and translation. And at the time of protein synthesis, what is happening? Genetic information required for the protein synthesis, which is stored in the DNA, first flows from DNA to what? mRNA. So this process is called transcription. Later, mRNA carries the genetic information required for protein synthesis in the form of codons, and these codons will code for amino acids at the time of protein synthesis from mRNA. This process is called translation. So, transcription plus translation results in the formation of a protein based on the genetic information present in DNA. This entire process is explained by Francis Crick, which is popularly called central dogma in molecular biology. So, simply speaking, what is central dogma? It explains the process of what? Direction of flow of genetic information. So what is the exact direction? At the time of protein synthesis, genetic information flows from DNA to mRNA. Later it flows from mRNA to protein, which is otherwise called a polypeptide. We understood what is central dogma, but there are few exceptions in living systems. Here I am showing an arrow mark, that is direction of flow of genetic information. It clearly tells you genetic information is flowing from DNA to DNA. Is it possible? Yes, it is possible. Which process makes it possible? That is nothing but DNA replication. Before this I was telling you, during the process of protein synthesis, genetic information flows from DNA to mRNA. But at the time of DNA replication, genetic information can flow from DNA to DNA. If you remember, what is replication? Synthesis of two identical daughter DNA molecules from the parental DNA, which occurs at the every round of cell division. So at the time of DNA replication, genetic information flows from DNA to DNA. That is also possible. And one more exception, let us see. that is reverse transcription. So reverse transcription is popularly called central dogma reverse, because just before I said what is transcription? Synthesis of single stranded RNA from double stranded DNA. But in very few RNA viruses like HIV and tobacco mosaic virus, etc., their genome itself is made up of single stranded RNA. So with the help of single stranded RNA inside the host cell, they synthesize double stranded viral DNA. This process is called reverse transcription, which occurs in presence of an enzyme called reverse transcriptase. So at the time of reverse transcription, genetic information flows from RNA to DNA. So students should concentrate on this. Usually at the time of protein synthesis, genetic information flows from DNA to mRNA, but genetic information can flow from DNA to DNA during a process called replication. And there is one more possibility that genetic information can flow from RNA to DNA. So it happens during a process called reverse transcription. So what is reverse transcription? Synthesis of double-stranded DNA from single-stranded RNA. Which enzyme makes it possible? Reverse transcriptase. That enzyme is also called RNA-dependent DNA polymerase. Okay. So in this video, let us discuss the first step of protein synthesis, that is transcription, in detail. So if you see here, I have shown a simple diagrammatic representation. This representation is of a transcriptional unit. So if you ask me, what is a transcriptional unit? It is the fragment of DNA which is copied as mRNA at the time of transcription. So students should have this clarity. At the time of transcription, entire DNA fragment is not copied as mRNA. So a small fragment of DNA is copied as mRNA. So that fragment of DNA which is copied as mRNA during the process of transcription is called transcriptional unit. So this represents a transcriptional unit. And if you ask me, what are the components of a transcriptional unit? So in a transcriptional unit, three important components are present. That is P for promoter, T for terminator. So in between promoter and terminator, what is present? A structural gene which is otherwise called cistron is present. So cistron is the fragment of DNA which is copied as mRNA. So such gene is called cistron. It is otherwise called structural gene. So if you look into the details here, I have drawn two strands of DNA. One strand running in 5' to 3' polarity, and the complementary strand is running in 3' to 5' polarity, and P stands for promoter. So if you ask me, what is a promoter? Promoter is the site where transcription begins. And once again, if you ask me a question, why transcription starts only at a promoter? My answer is, promoter is the binding site for the enzyme RNA polymerase, because transcription has to take place. And which enzyme makes it possible? There is an enzyme called RNA polymerase which helps in the synthesis of single-stranded mRNA on double-stranded DNA. So RNA polymerase cannot bind at random sites. RNA polymerase binds to a particular site that is called promoter. So that's why promoter is the binding site for enzyme RNA polymerase. That's why transcription always starts at what? Promoter. And here you can see we have terminator. Name itself indicates what is going to get terminated here. The synthesis of single-stranded mRNA is going to terminate at a particular site, and this site acts as terminator because it acts as a binding site for a special protein factor that is called Rho factor. So Rho factor helps in termination of transcription. So Rho factor binds to this particular site called terminator. That's why it is the site which helps in termination of transcription, and it is the site where transcription begins. In between promoter and terminator, what is present? Cistron, that is structural gene is present. Actually, it is the fragment of DNA which is going to be copied as mRNA in presence of which enzyme? In presence of an enzyme called RNA polymerase. It is otherwise called DNA dependent RNA polymerase, because in transcription what is happening? RNA is synthesized. What is existing? DNA is existing. Depending on existing DNA fragment, this enzyme synthesizes single stranded mRNA. That's why normal RNA polymerase is also called DNA dependent RNA polymerase. Till now we understood the structure of a transcriptional unit. Let me repeat, what is a transcriptional unit? A fragment of DNA which is copied as mRNA at the time of transcription. And here students should understand the two main differences between DNA replication and transcription. In DNA replication, entire DNA strand, entire parental DNA strand is copied as new DNA strand. But at the time of transcription, entire parental DNA strand is not copied. A small fragment of parental DNA strand is copied as mRNA. That is one point. And one more point is that at the time of DNA replication, new DNA strands are synthesized on both the parental DNA strands. But at the time of transcription, out of the two parental DNA strands, only one strand acts as a template strand. So with this it is very clear. What is transcription exactly? Synthesis of single-stranded mRNA on double-stranded DNA. That's why both the parental DNA strands do not act as template strand. Means at the time of transcription, only the 3' to 5' DNA strand acts as a template. With this it is very clear. mRNA is going to be synthesized on 3' to 5' DNA strand, but not on 5' to 3' DNA strand. If you ask me this question, why? It's very simple. RNA polymerase enzyme, which is also called transcriptase, always synthesizes mRNA in which polarity? 5' to 3' polarity. So as RNA polymerase can synthesize mRNA in 5' to 3' polarity, which parental DNA strand acts as a template? That is 3' to 5' strand. So students should understand this point. At the time of transcription, out of the two parental DNA strands, only one DNA strand acts as a template. Which strand it is? That is 3' to 5' strand. Why? Because RNA polymerase always synthesizes mRNA strand in which polarity? 5' to 3' polarity. So 3' to 5' DNA strand is template strand, and ultimately 5' to 3' strand is called non-template strand, and one more name it has. Template strand is otherwise called non-coding strand, whereas non-template strand is called coding strand. So here only many students get the doubt. Why do we call like this? It is very clear. Let me consider this strand, 3' to 5' strand is acting as template, right? Now, on 3' to 5' strand, nucleotide sequence is like this, AT, and what is the complementary sequence for this? Adenine always pairs with thymine, thymine pairs with adenine, once again thymine, adenine, guanine pairs with cytosine. And if you see here, at the time of transcription, when an mRNA is synthesized, nucleotide sequence on mRNA resembles the nucleotide sequence present on 5' to 3' strand, but not the nucleotide sequence present on 3' to 5' strand. So with it is very clear, nucleotide sequence present on 5' to 3' is coding for a protein. That's why 5' to 3' DNA strand is called coding strand, and 3' to 5' DNA strand is called non-coding strand. So it is all possible due to the presence of an enzyme called RNA polymerase, which is otherwise called transcriptase, because it is the enzyme which makes the process of transcription possible, and it is also called, what I said, DNA dependent RNA polymerase. So I repeat, template strand is a 3' to 5' strand, which is also called non-coding strand, whereas 5' to 3' DNA strand do not act as a template for the synthesis of mRNA. That's why it is called non-template strand. But non-template DNA strand is also called coding strand. We understood the difference between the template and non-template strand. When we go into the mechanism, I already told you. P stands for promoter, and promoter is the site where transcription starts. And if you ask me why transcription starts only at a promoter, because promoter is the binding site for whom? For the enzyme RNA polymerase. And here we have to remember one special name, that is sigma factor. Sigma factor is popularly called initiation factor for transcription. And why sigma factor is called initiation factor for transcription? Sigma factor helps RNA polymerase enzyme to recognize the promoter. With this it is very clear. RNA polymerase enzyme alone cannot recognize the promoter. So when sigma factor binds with RNA polymerase, together it is called a core enzyme, and sigma factor help RNA polymerase to recognize the promoter and to bind with it. Once RNA polymerase binds with the promoter, and this is the template strand, and on the template strand what is going to be synthesized now? An mRNA is synthesized in which polarity? 5' to 3' polarity, and sequence is always going to be complementary like this. But the only difference, if you remember, in RNA, in place of thymine what is present? Uracil is present. With that difference, an mRNA strand in 5' to 3' polarity is going to be synthesized on template DNA strand in presence of an enzyme called RNA polymerase. And once mRNA strand is synthesized, so this is the fragment of DNA which is copied as mRNA. Once this fragment of DNA is copied as mRNA, this process, that is elongation of mRNA strand, should be terminated, and this termination is the function of a special protein factor I told you, that is rho factor. So rho factor comes and binds to what? Terminator region. So once the rho factor binds with the terminator region, RNA polymerase enzyme cannot cross the rho factor. As RNA polymerase is unable to cross the rho factor, it cannot elongate the mRNA strand further. So that is how elongation of an mRNA strand in 5' to 3' polarity is going to be terminated. So ultimately, at the end of this entire process, what is synthesized now? A single stranded mRNA. In which polarity it is synthesized? From 5' to 3' polarity. And nucleotide sequence on mRNA is going to resemble what I said? 5' to 3' DNA strand. That's why it is called coding strand. And the only difference is that in place of thymine, here which base is going to be present? Uracil is going to be present. If I consider the same sequence like this, ATG is the nucleotide sequence on template strand. Now on the newly synthesized mRNA, what is going to be the sequence now? Here is adenine. In place of, in DNA thymine is present, but in RNA it is uracil, adenine, uracil, once again adenine and cytosine. So this is the sequence on newly synthesized mRNA, and this newly synthesized mRNA or the nascent mRNA is called a primary transcript. So what is primary transcript? A nascent or newly synthesized mRNA immediately at the end of transcription is called a primary transcript, and this primary transcript is subjected to little modification, and after the modification it gets ready for the process called translation, so which occurs on ribosome. So with this I want students to remember, transcription is what? Synthesis of single stranded messenger RNA on double stranded DNA. Transcription always starts at a special site called promoter, because Promoter is the binding site for the enzyme RNA polymerase. And who helps RNA polymerase to recognize the promoter? A special factor called sigma factor. That's why sigma factor is called initiation factor for transcription. And which protein factor helps in termination of transcription? That is Rho factor. That's why Rho factor is called termination factor. So at the end of the termination, what is synthesized? A nascent mRNA is synthesized, which is called primary transcript. So primary transcript after little modification gets ready for the next process, that is translation. And some important points about transcription given in textbook. They ask the question like this. At the time of transcription, if both the DNA strands act as template, what is the problem? It's very clear. If both the DNA strands act as template, two single-stranded mRNAs are synthesized. They will have complementary sequence, and once they form the hydrogen bonds, double-stranded RNA is going to be formed. And always students should remember, double-stranded RNA never participates in translation. For the process of translation, always what is required? Single-stranded RNA is required. So this point is very clear. At the time of transcription, if both the parental DNA strands act as template, what is the consequence? Two single-stranded mRNAs with complementary sequence are formed, which results in the formation of double-stranded RNA, which is never helpful or never participates in the process of translation. So protein can be never synthesized. That's why at the time of transcription, only one parental DNA strand, which strand? 3' to 5' strand only acts as a template strand on which a single-stranded mRNA is synthesized. So in this video, I have explained you what is central dogma, who proposed central dogma, Francis Crick, and what is central dogma reverse. It is nothing but the process of reverse transcription. Naturally, in biological systems, genetic information flows from DNA to mRNA, mRNA to protein, and this is the direction of flow of genetic information explained by Francis Crick. And in the process of protein synthesis, what are the two crucial steps? Transcription and translation. In this video, we have studied transcription. Thank you.